Aloha everybody. Last time we finished with biosynthesis of fatty acids. Today I will talk about biosynthesis of triglycerides or triacylglycerols, which is just another name for triglycerides. And then I will go over the biosynthesis of membrane lipids. After that I will talk about the biosynthesis of cholesterol, and we will finish with a discussion on lipoproteins. All right, so an interesting thing here with the way I'm presenting, at least, is that normally, like in many cases before, I have designed these slides to be interactive. In other words, I start with a question and then with an answers. Then the answers will be shown. But that's okay. I'm just going to present what I intend to ask you, and then give you the answers right away. So it's almost like asking rhetorical questions. So what is the primary use of fatty acids? When you think about primary use, meaning the most basic foundational overall use of fatty acids, and that is for energy. They're the best way to store energy, and I've said that many times before. Other use for fatty acids is for membranes. All cells are cells because they're organized, or they're separated from everything else by a membrane, which is made primarily of lipids. And also, fats are also used for synthesis of bioactive molecules. So over time, the use of fats obviously changes. I mean, for a growing organism that continuously increases in size, the demand of fats and energy for that matter for growth is increased. But once we reach a certain stage, that demand decreases, and it gets, the energy use gets repurposed to a point. And I usually try to make a funny point here that if you haven't seen that yet, or if you haven't felt it, I'm sure you will once you get into your 30s and 40s. You will see how suddenly your diet hasn't changed, but your shape has changed. Even though you haven't increased in height, you just suddenly start increasing, your width is starting to increase. So with growing bodies and growing organisms, highly functional organisms, the demand for energy is higher. But once things start to reach a certain point, and the demand for that energy decreases, the energy storage starts to increase if you don't adjust the amount of food intake. So if we take fat out of our diet, would that decrease the weight increase with age? Well, the answer really is yes and no, and that depends on the type of diet that you have and the quantity that you ingest. So, but I've made this point before, I don't want to go back to it. Ingestion of fat by itself, even though they're caloric, highly caloric, doesn't necessarily mean that it would contribute to a weight increase. That will depend on the amount of carbohydrates consumed with it, and how saturated our cells are with carbohydrates, because carbohydrates are the primary source of energy for the brain and many tissues. Fats are primary source of energy for liver, for heart, for resting muscles, and for the heart. But that doesn't mean that they don't use, whether the heart and the other muscles don't use glucose. They do. It's just that they're the primary use, like at resting state. So we also know that you cannot make glucose from fatty acids, but you can't make fatty acids from glucose. And we're going to make a point about that today again. And if you remember from last time, what is the building block of fatty acids? Acetylcholine. And what do we use to make acetylcholine? Carbohydrates and the carbon chains of amino acids, but primarily carbohydrates. So you don't need to eat fat to make fat in your body. All you have to do is continuously consume glucose in higher quantities than you need. All right, but enough with dietary related discussions. Let's just continue to the specifics of the biosynthesis of triglycerides or triacylglycerols. So what do we need for triglyceride synthesis? We need fatty acids and glycerol. Or, to be more specific, activated fatty acids and phosphorylated glycerol. Where do we get the fatty acids? We get them from acetyl-CoA. That's how we make fatty acids. Or we get ready fatty acids from food. And when are we going to use fatty acids from food? If our cells are, well, they have sufficient amount of energy from glucose, and then any ingested fats that we have taken in will simply be repackaged and sent for storage. What is the source of glycerol? Glucose and lipids from food. Remember, lipids do contain glucose. Triglycerides have a glycerol backbone. So from glucose, you can see shown here at the time when we reach dihydroxyacetone phosphate in the glycolysis pathway, then glycerol triphosphate dehydrogenase can reduce dihydroxyacetone phosphate to glycerol triphosphate. Also from food, the reduced glycerol from triglyceride or from lipid degradation and triglyceride degradation can be phosphorylated by glycerol kinase with the help of ATP to form glycerol triphosphate. All right, the resulting glycerol triphosphate is then acylated with two fatty acyl groups, forming phosphatidic acid. If you remember that from 481, basically phosphatidic acid is diacylglycerol with a phosphate as a head group. Before that though, as you can see on this figure, the fatty acids have to be activated. You cannot just add a fatty acid to the glycerol triphosphate. They must be activated in order to be prepared for the addition reaction. And how does this activation happen? An enzyme that you're familiar with, acyl-CoA synthetase, converts a fatty acid to fatty acyl-CoA, and then the activated now fatty acids can be added either at the first or second carbon of the glycerol triphosphate molecule. The resulting product is phosphatidic acid. Now, why did they say that you're familiar with this enzyme acyl-CoA synthetase? Because that is the same enzyme that activates fatty acids before they're attached to carnitine and transported into the mitochondria for degradation. It's the same process. It's the same enzyme. So you can see that enzyme activates fatty acids, and they can either be used for degradation or synthesis of triglycerides, or membrane synthesis for that matter. So it's the same enzyme that can be used in both situations, whether for oxidation or for storage. And the reason for that is because the purpose of this reaction is to activate the fatty acids for consequent reactions, whether that would be the addition of carnitine or whether that would be the addition of them to the glycerol triphosphate. It doesn't really matter. They need to be activated for the consequent reaction. Now, after we generate phosphatidic acid, that now is a precursor either for generation of membrane lipids, and here in this figure is shown the addition of a head group to the phosphatidic acid and generating phosphoglycerides or glycerophospholipids. We cannot generate sphingolipids from phosphatidic acid, so we can only generate phosphoglycerides. Now, before we make triacylglycerides, we need to dephosphorylate phosphatidic acid, basically forming diacylglycerol, and then add another fatty acid to now the hydroxyl group or the third carbon, I should say, and then we form triglycerides or triacylglycerols. All right, now that's basically about triglycerides. Now let's move towards membrane lipids, more specifically, membrane lipids. And the examples that we're going to give are primarily with phospholipids, but the processes for the membrane lipids that do not have a phosphate heat group, we're not going to really talk about that in this presentation. I'm not sure why is it not mentioned in the textbook, but we're just primarily going to focus on membrane phospholipids. Alright, so there are two major types of membrane phospholipids. If you remember, glycerophospholipids and sphingolipids. Now, how are they structurally similar and different? Now, here it's going to be important for you to remember these differences, even though it's a little bit removed. For 81, it's been a few months since you've taken, or for some perhaps even longer. So it might be good if you refresh your memory, but discussing them though will help you refresh that memory. So structurally at this point, briefly put, the differences are in the backbone and how many fatty acyl groups are added to the backbone. Now the backbone of glycerophospholipids is glycerol, which has 3 carbons, and then to it we have 2 fatty acyl groups added, bound usually by a phosphodiester bond, and then we have a phosphate-containing head group. Now the sphingolipids, they may appear similar in shape and structure, but structurally they're very different. The backbone is actually sphingosine, which is a long carbon chain, amino alcohol, and that it's not shown here, and then I'll show you a little bit later. But that sphingosine has attached to its second carbon via amide linkage one acyl group, and the head groups of sphingolipids can either be a phosphate-containing head groups, or they don't have to contain a phosphate. So there's a difference there. But the biggest difference again is in the backbone, and the way the acyl group is linked to the backbone is not via phosphodiester linkage, but through an amide or amide linkage, depends how you want to pronounce it. Okay, so in principle though, the synthesis of membrane lipids follows this outline. First, the backbone is synthesized, whether it's going to be glycerol or sphingosine. Then the fatty acids will be attached to the backbone, and then the hydrophilic head groups will be added. And the addition of these head groups could be done in the following 2 different ways, and we're talking about phosphor-containing head groups. That addition involves the help of a nucleotide, CTP. CDP in this case is added either to the diacylglycerol or to the head group. And then, regardless of whether it's added to the diacylglycerol or to the head group, the product would be a phosphate-containing head group, and CMP will be released. So the nucleotide in this case, as usual, plays the role of an activator. Now let's look at the biosynthesis of sphingolipids. So it begins with activated palmitic acid or palmitoyl-CoA, and then a serine is added to the activated palmitic acid, and the product is reduced to form sphingonine. Now you can see now that we have a long carbon chain amino alcohol. So sphingosine is an amino alcohol, and that is because we added serine, which has an amino group, to a long chain fatty acid, which is palmitic acid, 16 carbons. And you can see right here that the carbon chain is uninterrupted, even though it's shown as if there is a kink somehow here. But this is a continuous carbon chain. And here we don't have a hydroxyl group available to receive another acyl group, but an amino group. So after we form sphingosine, then we add a fatty acid to it to form an acyl sphingosine. The addition of the fatty group, the fatty acyl group, is done by an amide or amide linkage. I'm going to start using just amide as a pronunciation. So amide linkage, and then this is followed by desaturation, and we form ceramide. Ceramide is the precursor of all sphingolipids. From there, we can add either phosphorus-containing head groups like phosphatidylcholine and forming sphingomyelin, or we can add carbohydrate, oligosaccharides, or monosaccharides to form cerebrosides or globosides if we have a larger number of moieties attached to the as head groups. So this is how we generate sphingolipids, and primarily the phosphate-containing sphingolipids, even though here we show how we generate sphingolipids that do not contain a phosphate group. All right, now we will transition to the biosynthesis of cholesterol. Now, even though cholesterol is essential for the body, it is not an essential food group. How is cholesterol essential? On this figure you can see, and I've mentioned that before, that cholesterol is the precursor molecule for synthesizing steroid hormones, bile acids, and vitamin D. Vitamin D generation is actually, it happens with the help of UV light in your skin. And so that's why it's important for you to be exposed to regular sunlight, of course, in a balanced way, not to burn yourself, but not to hide yourself continuously, because vitamin D is important for a proper development of your bones. Bile acids for proper digestion of fats, and steroid hormones, obviously they have a various function, but it's essential for the development of our bodies. Now, the process of biosynthesis of cholesterol is deemed as one of the most complicated synthetic pathways. We're going to try to simplify it and make it bearable, and actually it is fairly bearable, so I don't want you to dread it. But you should be aware that the building block of cholesterol, the foundational building block, is acetyl-CoA or acetate groups. These acetate groups are used to generate isoprenes. The isoprenes, or isoprenoids, activated isoprenes, which are shown right here, are a molecule that is used for the synthesis of a lot of other bioactive molecules in the body. So the immediate precursor, or the immediate precursor for cholesterol is actually isoprene, but you will see that isoprenes are made by acetyl-CoA, which, again, where does it come from? From glycerol. So you generate cholesterol, not glycerol, glucose. You generate cholesterol from glucose. Again, that's why glucose is so essential for our bodies. You have to have it in order to generate all these molecules that are based on acetyl-CoA. Glucose generates acetyl-CoA in times of plenty. And again, why are we not using fats for that? Because when you supply the body with necessary amounts of glycerol, fatty acid degradation or fatty acid oxidation or beta oxidation is suppressed. And you either the focus on the body is on glycerol and generating acetyl-CoA that is burnt in the citric acid cycle to generate energy. And again, fatty acids, for the most part, in most cells, is suppressed, unless they're adapted to use fatty acids for their own energy. But under excess of acetyl-CoA, well, I shouldn't say excess. When acetyl-CoA is available, it can also be used it's also used, not can be used, but it's used for generating cholesterol as well. So such a foundational role for acetyl-CoA. Alright, so now let's look at the biosynthesis of cholesterol. Now we're going to break it into four major steps. The cholesterol synthesis, we're going to break it into four major steps. The first one is condensation of three acetyl-CoAs or acetate groups into the formation of mevalonate. Then we'll convert mevalonate to activated isoprenes. There are two types of activated isoprenes. Then we will take 6 of these activated isoprenes and we will condense them into forming of this much larger molecule called squalene. And then the last stage is the cyclization of squalene to form cholesterol. Alright, so let's look at the first stage. We have 2 molecules of acetyl-CoA. With the help of an enzyme that we're already familiar with, we form acetyl-acetyl-CoA. Then we add another molecule of acetyl-CoA to form HMG-CoA. Are you familiar with this? You should be familiar with this. This is basically the beginning of the synthesis of ketone bodies. So it's this pathway follows uses the same enzymes used by ketone body synthesis. However, from here on, things are different. Instead of removal of the acetyl-CoA from HMG-CoA and the formation of acetoacetate, which is, I guess, the first one of the ketone bodies, we reduce HMG-CoA to mevalonate. So, first stage completed, we have mevalonate. The next one is the conversion of mevalonate to activated isoprenes, and I want you to notice the role of phosphorylation. So we have mevalonate, one phosphorylation, second phosphorylation, third phosphorylation. Okay, so we have a pyrophosphate and we have another phosphate added. So whenever phosphates are added, these are excellent leaving groups, whether it's pyrophosphate or phosphate. So you're preparing the molecule for something, for reactions of some form. Usually, well, those are activating additions. That means that carbon that is bound to these groups is activated for consequent reactions, usually for some form of addition. Alright, so what happens after the 3 phosphate group additions? We have decarboxylation and an inorganic phosphate release, and now we have these 2 forms of isoprenes, either the double bond here or in this position or in this position, regardless of where it is. Obviously, we're preparing this for interaction, for some form of condensation reaction. I mean, this is an electrophilic carbon. This is going to be a reaction here. This is an excellent leaving group. That means that this carbon is also activated. I mean, this is a fairly well activated molecule. So there's going to be we're preparing this molecule for the follow-up, which is basically condensation reactions. So we get the 2 types of activated isoprenes and we fuse them together. Surprise, surprise. And we form geranyl pyrophosphate, which is still activated. The geranyl pyrophosphate will be used to add another molecule of activated isoprene. So far we get 3 activated isoprenes to form farnesyl pyrophosphate. And then 2 farnesyl pyrophosphate molecules are fused together to form squalene. So the total number of activated isoprenes is 6. We use 2 to make geranyl, then we add another one to form farnesyl. So each farnesyl is made out of 3 activated isoprenes, and then we fuse 2 farnesyl groups together for a total of 6 activated isoprenes used to form squalene. The third stage is completed, and then we basically will use that open hydrocarbon chain and close it, cyclicize it, and form the typical ring structure of all cholesterol, shared by all cholesterol molecules. So we show here stigmasterol, inosterol, ergosterol, and of course cholesterol. And I'm asking what is common between all steroids. Well, notice the ring structure is fairly similar. Whether you have multiple double bonds or not, it doesn't really matter. You have the 3 6-membered ring followed by a 5-membered ring, a very typical ring structure of all. Something else that is similar, notice they have a hydroxyl group as head group, and then all of them also have an aliphatic tail. And that's typical for all of them. Remember, these molecules will be included in the membrane, and the membrane molecules always have a hydrophilic head group and a hydrophobic tail. This is a very rigid molecule, and whenever it's inserted in the membrane, we've talked about this in 481, it adds rigidity in the membrane as well. So, this is it for biosynthesis of cholesterol. I promise you that we're going to make it bearable. So basically all I want you to know is the different steps. You don't try to remember the name of the enzymes, just know what happens. Alright, so now I'm going to focus on the last stage of my presentation, which is what it says here, biosynthesis of cholesterol. But the only reason why it's called biosynthesis of cholesterol is because lipoproteins are usually mentioned, or we are aware of them, because of atherosclerosis and their role in the formation of atherosclerosis, which is a major reason for cardiovascular diseases, or cardiovascular disease, or any type of vascular disease, whether it's in the brain or whether it's in the heart, it doesn't really matter. Whenever we have a hardening of the vessels or them losing their elasticity, we blame lipoproteins, or specific kinds of lipoproteins that are rich on cholesterol. So, how do we transport cholesterol and other lipids in the body? That's done through these special complexes known as lipoproteins. Remember, lipids are hydrophobic. They cannot mix with the hydrophilic environment of the cell. So they're made out of proteins, and they're made out of lipids and proteins as well. They encapsulate the triglycerides or anything else that is primarily hydrophobic, almost as in a micelle or cell-like looking structure. It also involves proteins. These are known as apolipoproteins, and as a total, the structure is known as lipoproteins. They have a different size and different roles. Here you can see images of these structures. The largest of those is kilomicrons, then the VLDLs, LDLs, and HDLs. In the next slide I will explain that in greater detail. Alright, so lipoproteins have, as seen here, various composition. Obviously they're greatly different in size and in function. Whenever we talk about lipoproteins, we usually talk about density. And here's something that could be a little bit confusing. because when we see large ones, we usually say, oh, large density. When we see small ones, we always small density. Actually, it's reverse. Whenever we talk about density, we're talking about the relative concentration of protein. You can visualize the density basically thinking about it. If you imagine, even though it's not exactly the case, if you imagine that these different sized particles contain the same total number of proteins, you can imagine that if you add more fat to that particle and blow it up in size, the concentration or the proportion of proteins to fat is going to be much smaller here in larger particles because he has so much fat compared to these tiny particles, which are primarily, well, it have very little fat in them, and so the proportion of protein to fat is much higher here than here. That's why HDLs are known as high density lipoproteins, because they have relatively more protein with respect to fat, and VLDLs are very low density lipoproteins because the ratio of protein to fat is much smaller since they contain a lot more fat. And we don't even talk about, well, the density is not even included in the kilomicrons. Well, functionally they're a little bit different, and also they're very large. Okay, so let's look at the individual particles. Kilomicrons are the largest, least dense, and they carried lipids from the intestines to tissues and the liver. They are the lipoproteins formed in the intestines, and then they get, that's how the fat is being transported from the intestines primarily to the liver, but also to the muscles and other tissues. Actually, I want to point out this table. Just look at the the protein percent. This is not an actual value. Well, it's an actual value, but it's a percent. It's like a ratio. It's not an actual number. In other words, this doesn't say that each particle contains, for example, 200 protein molecules. They tell you the ratio or the proportion of protein to fat. And you can see how the density or the percent protein as part of these particles increases, going from kilomicrons to HDLs. All right, so kilomicrons move fats and cholesterol from the intestines to the liver primarily, but also to other tissues. Now the liver will pick up the kilomicrons, take the cholesterol and all the fats that they carry, use them for whatever purposes you use them, but usually if there's no glucose and the metabolism is focused on fat, then the liver will make sure that the fat is used for that, for energy. But if we have plenty of glucose, the liver will basically package the fat and send it out for depositing for future needs. So regardless of what the use is, though, after the lipids enter the liver, they will be repackaged into different types of lipoproteins, and they are the very low density lipoproteins. These are the VLDLs. Now the VLDLs, like it says here, they're generated in the liver, and they're rich on triglycerides. They also contain cholesterol, but their purpose is to transport now these lipids to the tissues, muscle, adipose tissue, whatever or whenever this is needed. That's the purpose of VLDLs. And the lipoproteins now have the responsibility to contact corresponding lipases on the endothelial on the surface of the capillaries and small blood vessels, and exchange that or donate, deliver that fat to those cells, and then from there they get delivered to the corresponding cells that would use the lipids for whatever purposes they are, whether for storage or for energy. And so you can imagine the VLDLs, they're like delivery tools, and they slowly offload primarily triglycerides. All right, but if you imagine these particles, the more fat they donate to the target tissues, the smaller they will get. And they primarily deliver triglycerides. And then the more they deliver, the smaller they get, and at some point their size becomes the size of the next class of lipoproteins known as LDLs, or low-density lipoproteins. They are basically triglycerol-depleted VLDLs, and they are rich on cholesterol. Their purpose is to deliver cholesterol to the extrahepatic, or all the other tissues other than the liver. And the LDLs are also labeled on your blood test as bad cholesterol. And because if you have high LDLs, that usually puts you at risk, or is considered to put you at risk, and it is, for development of atherosclerosis. I'm going to talk about this a little bit later. So that's why they are labeled LDLs. There is another class of lipoproteins. They are known as HDLs, or high-density lipoproteins. These are liver-generated intestines. I'm reading intestines here and misspoke. They're usually generated in the liver and also in the intestines, but their purpose is, I call them, I don't know whether other people call them like that, but I call them as scavenger lipoproteins. Their role is to pick up remaining cholesterol from lipoproteins or other tissues, take it and deliver it back to the liver for repurposing. They are known as the reverse cholesterol transport. So these lipoproteins on your blood tests are called the good cholesterol. And the reason why it's called the good cholesterol is because, again, their purpose is to pick up excess cholesterol remaining from LDLs, VLDLs, or other tissues, and bring it back to the liver. And for that reason, it's believed that, if you have a higher concentration of those, you have a lower concentration of LDLs, and the possibility for hurting your blood vessels is decreased. However, this is a double-edged knife. This is still cholesterol, whether you have high HDLs or low HDLs. Obviously it's better to have more HDLs than LDLs, but still the total cholesterol still should be below what's considered a threshold or a dangerous level. So it's still part of total cholesterol, what I mean to say. All right, here I'm just showing you a table. I don't have any intentions for you to memorize these. Just wanted to show you that the lipoproteins have a signaling role. These apolipoproteins that are part of the lipoproteins, they're signaling molecules. They help the lipoproteins to anchor and target specific tissues and cells, and that's how they deliver their load to whoever is intended to receive it. So these are signaling molecules, and it's very important. They're part of these molecules. If you have mutations in these molecules, the lipoproteins can no longer function properly, and people that have faulty or changed apolipoproteins there is a so-called familial atherosclerosis or familial hypercholesterolemia, where you have very high cholesterol in the blood, even though your diet is not rich on cholesterol. And that is because there is a problem with the offload of cholesterol from the lipoproteins. As a matter of fact, 20-some years ago when I was in medical school, my professor in pathophysiology told us that it's not, the atherosclerosis is not a simple, it's not a simple function of how much cholesterol you consume. He said you can consume cholesterol and you might not have it, and you don't have to consume cholesterol and you can have high cholesterol in the blood. Because in some people it's about how the cholesterol is transported in the body. And he says nobody develops atherosclerosis by eating eggs, which are the richest food group on cholesterol. I mean, eggs, the egg yolks are extremely rich on cholesterol, but nobody would develop atherosclerosis by eating eggs. Now he says, and I agree with that, and I've seen it in many cases here in the United States, especially because eggs are, promoted as an excellent breakfast food, and many of us, including myself, love eggs. However, I was taught from a little child that you don't eat a lot of eggs. You can eat a few eggs a week, but not too many. Where I've seen at least some of my friends here, they eat every day for years, even decades. Every day they eat a few eggs for breakfast. Well, not surprisingly, many of these friends that I have lost their gallbladders in their 20s or 30s. And for me, it was such a surprise to see such young people losing their gallbladders because of, for whatever reason. And I attribute that to high cholesterol in your diet. Because if you remember, cholesterol is used for the synthesis of bile salts. And where do they get deposited? In the gallbladder. And if you have too much of it, they conglomerate and they form gallbladder stones. And once you start making those, well, eventually you're gonna get in trouble with it. And if that gallbladder gets inflamed, then you get a surgery and you get your gallbladder removed. So what my professor was saying at the time, it's not so much about how much cholesterol you consume. You can eat eggs a lot. You're not going to develop atherosclerosis, but you will develop gallbladder stones, most likely. Especially if you combine it with butter or with other fatty meat, like bacon or sausages, then you're basically a recipe for gallbladder removal in your 20s, 30s, or 40s for that matter. All right, so in order for you to develop atherosclerosis, there are usually problems with the apolipoprotein signaling, which does not allow for proper exchange of that load that LDLs have. They cannot offload the cholesterol, and so they start making, which let me get to this slide, they start getting deposited in the capillaries, and they harden capillaries, especially in the area with a lot of friction. And so also if the endothelium of the capillaries is harmed, these lipoproteins tend to wedge there. They start building up. Calcium starts depositing there as well, so it hardens that area. The area starts to bulge a little bit and starts growing, and at some point the opening of the capillary becomes smaller and smaller and smaller, and you can experience, or the person can experience, problems with proper blood supply in the tissues post that thickening or obstruction. And the worst thing happens when that plaque, if it's softer, especially in the core, and it happens to burst, the bursting itself might not be the biggest issue, but because it bursts, then what happens is that the blood clotting system gets activated, and a blood clot gets formed right there at that plaque very fast, and then you develop either a stroke or heart attack, because now the clot that is formed there completely obstructs the blood vessel, and the blood does not pass to the following tissues. So that's the danger of atherosclerosis, is that thickening of the blood vessels, and then the resulting complications either for the heart or for the brain, because these are usually the most common complications, because the blood supply of these two organs is incredibly important. So here, though, just to generalize, kilomicrons from the intestines, they get in the blood and they go back to the liver. The liver repackages the lipids and synthesizes VLDLs. The VLDLs go back into the blood supply, and then they offload the triglycerides to the tissues, and they slowly get converted to LDLs. The LDLs continue to offload cholesterol, and some of them, or all of them actually, eventually get received back by the liver. But if these LDL continues to circulate in high concentration, that's where atherosclerosis forms. HDLs are also generated by the liver. They're in the blood supply with their purpose to pick up cholesterol from remnants of VLDLs and LDLs and deliver it back to the liver for further processing. All right, I hope this was informative. I hope things made sense. With that, I will conclude my presentation. If you have any questions or anything, if something is unclear, please let me know. Come and see me during my office hours or email me. Thank you for your time, and until our next time.